Alrighty, so as you can see, we are going to be going through photosynthesis details. Okay, so just the first thing, probably just nice and basic, right? In that different kinds of organisms do photosynthesis, of course. Most of the stuff that we're talking about has to do with plants doing photosynthesis. But others, of course, do that, including some protists like euglenids or euglena is an example of that, right? And some bacteria like cyanobacteria, like sometimes they call it blue-green algae, right? And stuff. It's not actually algae. It's actually like green pond scum is like cyanobacteria. So the big thing is with photosynthesis, of course, you're absorbing this light energy to get the energy source to do the process. So we need to know a little bit about light because of that, right? So light's weird in that it acts as a wave and a particle. So the wave it's electromagnetic energy, right? In other words, the radiation you get from that, okay? Versus it's also a photon, a little Star Trek photon torpedo kind of a thing, right? All right, but what we're interested in is that electromagnetic spectrum. In other words, kind of like it's the fancy name for just like, the rainbow and all the things that are on either side. So it ranges from the gamma rays, like gamma radiation, thank you Hulk, right, to those long radio waves. So when you're doing, that's what they mean by the carrier wave for radio, when you take in and get that signal. The one that we're interested in when it comes to photosynthesis, though, is that really small band, okay, with nanometers there, from 380 to 725 nanometers of that visible light. So what we kind of talk about being white light. But of course, as you know, when you look at white light through a prism or like through the prism that gets formed through rain, you're going to get that rainbow. And so it's that we're interested in. The other thing is looking at what kind of thing that you need for doing photosynthesis. What parts do you need? Where does it happen? And since we're talking about plants, what we're interested in, of course, is this leaf, okay? So the leaf itself, when we're looking at it, right, here's the top and the bottom of the leaf. There are the holes in the bottom of the leaf. That's the stoma. So that's a hole, just like, your pupil is a hole. And just like you have an iris that opens and closes your pupil, you have guard cells that open and close that stomata, or stoma. Okay, stomata is singular, stoma is plural. So it just means hole. And the big thing is that's where you're getting gas exchange, because photosynthesis needs gas. They need CO2, just like we need oxygen, right? They need to be able to exchange gases, and they don't have, a mouth to do that, but they have these stomatas or stomas to do that. So that's why there's this spongy mesophyll that's right up above it. That's where it's exchanging, that's why it's spongy, so it can store all of these extra gases. You can get carbon dioxide in, oxygen out. The problem with that is that sometimes that means you're going to have your stoma or stomata open when it's hot, and you're going to lose water. So that can be a problem for certain plants. That's why the majority of plants that you're looking at are C3 plants and stuff, but there are what we call C4 plants and CAM plants that we're going to talk about later on in the notes, okay? So there's also the palisade mesophyll. So it's palisade like a palace has columns, right? These are columnar cells. This is where most of photosynthesis occurs. And the spongy mesophyll is where a lot of the gas exchange occurs. And then, of course, there's a cuticle on the surface. That's a waxy coating, just like the cuticle, that's here at the end of your nail. That's a waxy coating that helps reduce water loss or makes them waterproof. It's like in a rainforest, things like that. And then you have these epidermal cells that don't do much, just like you have epidermal cells. And below that, you have dermis, that does a lot of the jobs of like hair growth and sweat glands and all that. So key parts to know, of course, are the cuticle, that waxy coating for water retention and waterproofing, your epidermis, which is the surface, it's protection as well. Veins have the xylem and phloem for transport of water and sugars. Xylem and that's what the X is here, is that xylem transport water, phloem's just the sugars. We'll talk more about that when we do some other stuff with plants later. Mesophyll is the middle tissues, right? So these are the ones, that's all meso means, is middle. So middle, right? Mesophyll is just middle filling. And that's why you have palisade, because they're the column ones, and spongy, because it's got holes in it like a sponge. Stomata being the holes, or the stomas, guard cells that regulate the size. And then there are what are called bundle sheath cells. Because in your diagram, I'm sure it's better than this, it has some veins in it, like when you look at a leaf and stuff that you have, like when you're drawing a leaf, when you were in kindergarten probably, and you draw this, and if you're like me, this is how you probably still. This is the venation of the leaf. That's where the xylem and phloem are. And around the xylem and phloem, there'll be what are called bundle sheath cells around it. That comes in when we talk about things like the C4 plants are going to be talking more, and CAM plants, you're going to be involving these bundle sheath cells. Okay, that's not on this diagram, but I'm sure it's on yours because in the diagram of 725, it has that. Okay, so let's look at the photosynthetic portion of that, and that is the mesophyll. So the mesophyll has, for the gas exchange, right, that's spongy. It has the palisade or columnar cells, right, which is where you've got these mesophyll cells that have chloroplasts in them, and those chloroplasts have a thylakoid membrane, so you got a membranous sac, which is where the actual light-dependent reaction, remember how we've talked about, right, how to be able to do something that has an electron transport chain, you've got to have a membrane and an itty-bitty living space. Remember that itty-bitty living space, right? So you need these membranous sacs and that space that's inside of them to be able to do these, okay? Which we'll get into on the next page. All right, so you got the thylakoid space, which is inside, right? That's an itty-bitty living space. You have the thylakoid membrane itself, which has, make sure on the left-hand side where your chloroplast is, all your thylakoid membrane, please color it in green, okay? Because that's where the actual pigment is. The chlorophyll A, the chlorophyll B, the carotenoids, all those are going to be there. Then you got the grana, right, which are going to be a whole bunch of these stacked in, it's like a pancake stack, okay? Then, of course, you've got, just like in the mitochondrion, where you have the goo that's in there, that's the matrix. In the chloroplast, that would be, of course, the stroma, that dense fluid with all sorts of enzymes, and it's where a cycle happens. So just like in aerobic cellular respiration, where the Krebs cycle occurs in the matrix, you have the Calvin cycle, the cycle again. All cycles are going to occur in the gooey stuff, in this case, the stroma. So let's talk about those pigments, right? Let's review those pigments. Those pigments include, right, certain jobs. Pigments, their job is absorption. So their molecules, it's just the basic definition is it's a molecule that absorbs any part of the light spectrum. So it could be a pigment like the carotenoids. So they're going to absorb in a different part of the light spectrum than, say, chlorophyll A and chlorophyll B. And in fact, right, if chlorophyll A and chlorophyll B look green to us, think about it this way, right? If they look green to us, what colors are they actually going to absorb? In all reality, they're going to absorb better, amazingly enough, right, at reds and blues, not green, right? Because if these things are green, that means they don't like that green. They reflect it back at you. They don't use the light. So the worst thing you can do is put your plant under a green grow light. If you ever notice, grow lights usually are red. They're that way for a reason, okay? Now, the light that is not absorbed is reflected or transmitted, hence the reason chloroplast and chlorophyll are green. They don't use it, so they give it back to you. So it's not useful to put it under there, okay? Types of pigments. There's chlorophyll A. That's the main photosynthetic pigment. That's why they put it with a name. There are two types. There's P680 and P700. And what that means is it's just describing what wavelength of light it absorbs most efficiently, okay? So remember how there's a photosystems 2 and a photosystems 1? At photosystems 2 has P680, photosystems 1 has P700, and that's why they're a slightly different green color, and they're absorbing slightly different wavelengths. Okay, so they're blue-green in color. They absorb this red and purple, so this is why if you want to have a grow light, you're going to get blues and reds, okay? And they're the ones that are in that reaction center of the photosystems. So remember when I had you do, I'm just going to go like this. Remember when I had you do this for the warm-up? This is the reaction center here in the middle. So this is where you're going to get the chlorophyll A P680 and the chlorophyll A P700. Okay? Now, There is also, of course, chlorophyll B. It's an accessory pigment, like an accessory to a crime. They're the ones who help the job, but they don't necessarily do it themselves. So it also absorbs light energy. They're lighter in color. They absorb different wavelengths of light, and their job is to transfer that energy of excitement through electrons to the chlorophyll A. So they are in the light harvesting complex of that photosystem. So there again, like we did on that one warm-up, you've got chlorophyll B is going to be here and here, okay, out in here, right, the chlorophyll B, and it's in this outside portion that's the light harvesting complex, okay, that light harvesting complex, okay? So their job is to hug, to hold, and to transfer all of their energy to the chlorophyll A that's here in the middle, okay? So, you also have the carotenoids. Ooh, got a little excited there, sorry. The carotenoids or carotenoids, I like to say carotenoids because it helps me remember their color. They're yellow and orange like carrots. They absorb better at different lights as well. And they also occur in that light harvesting complex of the photosystem proteins, okay? They act not only as an accessory pigment that transfers electrons and gets chlorophyll A more excited, but it also is natural sunscreen, okay? And so it's photoprotection, because just like our cells can get cancer if we get too much sunlight, like skin cancer, plants can also get cancer as well and have damaging of their cells. All right. So, light-dependent reactions, let's look in detail, because of course I know you want to go there, okay? So, light-dependent reactions are also known as photosystems 2 and photosystems 1. They're photosystems, the thing, that's the protein complex that's in that thylakoid membrane. That's the stuff that you drew, the thing, okay? Then, there's the structure, right, of that thing, the light harvesting complex that has the chlorophyll B and the carotenoids, okay? And it encircles, it goes around, it's kind of like, remember, it's like, a donut, okay, with a little yummy filling, okay? So you have your light harvesting, right, is all the way around the reaction center that's here in the middle, okay? And the general process that occurs there would include having these pigments collect the photons, sorry, that's awfully bright, collecting the photons that then transfer the energy by transferring electrons to the reaction center. So in other words, these little guys right here get all excited. When they get excited, they start flinging off electrons. Those electrons that are getting flung off then get transferred over here to chlorophyll A and get them doubly excited. Doesn't matter if it's a carotenoid or if it's chlorophyll A, they're also all pigments, when they get excited, fling electrons. They all get excited, start flinging off their electrons. And then they start transferring, and this one makes this one excited, and it flings off its electron, and then it starts giving its electron this way. So everybody gets excited here and passes on the love here to the middle with the chlorophyll A. Okay? Then you have the reaction center there in the middle that has that chlorophyll A, either P680 or P700, depending on whether it's photosystems 2 or photosystems 1. It also has that primary electron acceptor molecule bound in that same protein. That's why you get, on this picture, this little Eve-looking thing from Wall-E, right? Here's your primary acceptor molecule, and here's your chlorophyll A. They tend to be on the opposite ends of this chunk of protein that's embedded in the membrane. That's called that lovely reaction center, okay? So then, you've got it being surrounded by that light harvesting complex. So just like I did with this a minute ago, here's your reaction center, and here's the light harvesting complex that has the carotenoids and the chlorophyll B, and then in here is all the chlorophyll A, either P680 or P700, and it's surrounded by that light harvesting all the way around like this. A little hug of love. All right, so then what happens is that chlorophyll A receives energy in the form of either excitement from these electrons here, and light itself is going to get chlorophyll A excited. It then gets excited and releases its own electrons and transfers that electron to the primary electron acceptor molecule. In other words, these electrons, when these guys get excited, they go bing, and these little electrons go up to and bind with this primary electron acceptor molecule, which then passes it on to an electron transport chain that's nearby. Okay? So anytime a pigment gets excited, it flings off electrons. Okay? And that always needs to go somewhere, either to another molecule or to a primary electron acceptor molecule. So, process-wise, okay? Typical process is what we call a non-cyclic electron flow, okay? That means it goes from one thing to the next. In other words, it goes photosystems 2, yes, it's first, to photosystems 1. So, photosystems 2, chlorophyll A, P680 gets excited. It loses, it's so excited, it starts flinging it off. It's kind of like, I always kind of think of it as like some cheesy little biochemistry burlesque show. So you've got this chlorophyll A gets all excited, starts flinging off its little electrons, okay? Those then need to be stabilized. Chlorophyll A without its electron is very unstable. Therefore, it's going to take some electrons from water that's nearby and split that water molecule to steal an electron, which releases hydrogen ions, which are going to be helpful in doing that, remember the electrochemical gradient, where we're doing that photophosphorylation, you're going to need that. And then oxygen gas, thank goodness for us, because, well, we like to live, okay? And so this does a couple of things. It's beneficial to us. It gives them some oxygen to do their own cellular respiration, or aerobic cellular respiration. It gives some hydrogen ions out into that stroma so that you've got that available. And it stabilizes the chlorophyll A so it can be excited again and just be used again and again. So excited, fling an electron, get stabilized. Excited and get an electron, flings off and then get stabilized again. So you can use the same thing again and again. We're also going to go through a picture version of this on that one diagram here on the next video. All right. So hopefully this is actually recording, and anyway, it's really weird not to have you guys here for me to ask questions. Anyway, there you go. So the electrons, right, get transferred to the primary electron acceptor molecule. Then it passes to an electron transport chain, which just means chunk O proteins in the membrane that they pass it along. When that happens, just like in aerobic cellular respiration, remember how electron transport chains, those proteins get excited? Remember when we talked about this, right? So when we talked about this, and these little proteins get all excited, okay, with some electrons, it starts pumping hydrogen ions into this itty-bitty living space. Same thing. Hydrogens are pumped into itty-bitty living space of the thylakoid space, okay, from that stroma, just like in cellular respiration, where it went from the matrix goo into the itty-bitty living space of the intermembrane space. Okay? So then, that means you do the exact same stinking thing that you do in aerobic cellular respiration, chemiosmosis. You get photophosphorylation that causes an increase of hydrogen ions in that thylakoid space, that itty-bitty living space, right? It then creates a proton gradient, which is also known as an electrochemical gradient, also known as membrane potential, because you're getting that membrane to have the potential to do work. In this case, chemical work, okay? And the chemical work that needs to be done is ATP synthesis. So you have, just like in aerobic cellular respiration, you have ATP synthase allowing those hydrogen ions back across the membrane down their electrochemical concentration gradient. And when that happens and it goes back out through the membrane and into the stroma, energy is generated that helps convert the process of gluing together ADP and a phosphate to make ATP. This ATP is then going to be utilized, because we're talking photosynthesis now, it's going to be utilized to run the Calvin cycle. But that's not the only source of energy to get that Calvin cycle to run. Part of the Calvin cycle is what we call the reduction phase. So that's why photosystems 2 is happening after that. So photosynthesis 1 is happening after photosynthesis 2. In photosystems 1, you get chlorophyll A P700 this time, getting excited just like before. It's getting excited by light and electrons from the photosystems 2 electron transport chain. Okay? So you're getting ATP made, but you still got some electrons running around free. That's not good. It then gets pumped into photosystems 1, okay? And so chlorophyll A P700 gets doubly excited. But as with any pigment, when it gets excited, electrons pop off. Electrons that transfer to a primary electron acceptor, okay, molecule just like before, and goes into a different electron transport chain that produces NADPH. So what I'm going to do is I'm going to go over here and I'm going to grab a little picture that I use in IB12 for this part. So in other words, you have two photosystems. You have photosystems 1, it's right here, and photosystems 2. If everything is going what we call the non-cyclic flow, right, that you've just written down, that means photosystems 2 gets excited, okay? You have these guys, the chlorophyll B and the carotenoids getting excited, transferring electrons to chlorophyll A P680, that transfers an electron to the primary electron acceptor, that transfers it to an electron transport chain that is causing hydrogen ions, to go into this itty-bitty living space inside here. Those electrons get transferred, right? Hydrogens come in here, but the electrons get transferred to photosystems 1 that is going to have chlorophyll B and carotenoids that are going to then transfer electrons here to the chlorophyll A P700, which is going to lose an electron to the here, okay, to that primary electron acceptor, which is then going to go to a different electron transport chain that doesn't do any pumping, because its job is to help form NADPH plus H plus, which is an electron taxi, so it uses that electron to make that, okay? Over here, you don't need an electron to make ATP, okay, that's over here. But right over here with ATP synthase. But that electron is going to stabilize these molecules so you don't have to do photolysis. Remember back here, when we talked about stabilizing, right, where to go to an island? Where is it? In the reaction center. Okay, it gets released, right? Chlorophyll A receives, right? And so then what's going to happen is it's going to do photolysis during, right here, okay? Means that it's going to have some water molecules right here are going to get split. You're going to give off hydrogen ions. Oh, golly gee, once it can come into here. Some oxygen that gets used by oxidative phosphorylation and aerobic cellular respiration. And it yanks in the electron to stabilize these pigments again, okay? All right, hopefully that helped. Now, photosystems 1, though, can do something a little funky. It can do what's called cyclic electron flow. In other words, photosystems 1 runs all by itself. It self-stimulates, okay? What that means is, okay, you've got P700 getting excited, but instead of going into the electron transport chain right here, okay, instead of going over here and helping move on to the taxi of the NADPH plus H, what it does is that electron loops back and just runs a circular track like this. That's why it's called cyclical electron flow, okay? So it self-stimulates. It goes here, gets excited, thinks it's an electron, goes through here, and then it gets excited by its own electron again, okay? So that's the cyclical flow, in that it transfers it still to its same primary electron acceptor molecule, that piece at the top, right? That one that's right here, okay? But then what it does is it passes it to the electron transport chain of photosystem 2, and then it transfers it back to itself. So therefore, instead of doing the typical go through the process and make NADPH plus H to be the little electron taxi to the Calvin cycle to do that reduction phase, okay? It's going to do this, which is going to cause hydrogen ions to move in, which is going to cause electrochemical gradient build up here, which is going to cause them to want to go back out this way through ATP synthase, and so you're going to get chemiosmosis to make more ATP. So the plant can actually cause it to do that when more ATP is needed, okay, than NADPH. All right, so let's finish this up because I got to be to a meeting at 5. I'll come back and I'll do the one for the diagrams for the other one here when I come back after my meeting. All right, light-independent reactions, also known as the Calvin cycle, is where there's 3 phases. You've got carbon fixation, okay, which is where you're going to take a carbon off of that CO2. That is going to then make it attach to, okay, it's going to make it attach. I'm just grabbing another diagram from IB12. So, sorry, those of you IB11, you're going to be bored next year. So it takes, and here's CO2, it's going to take a CO2 off of this and attach it to what's called ribulose biphosphate, which is a C1,2,3,4,5 molecule. It uses an enzyme called RUBP carboxylase, because it's an enzyme that adds carbon to this molecule. It then makes an unstable 6-carbon molecule that as soon as it forms, it goes whoop, and pops it apart into some C3 molecules, okay, that you don't need to know the name of at this level. So, we'll talk more about that in IB12. Have fun! All right, the reduction, remember how I said some of those electrons, right, from that lovely electron taxi known as NADPH plus H plus, right, is where you've got some ATP coming in to phosphorylize. That's why there's this little thing right here. Phosphorylation adds energy to this 3-carbon molecule, okay? Then you've got the C3 molecule being reduced because NADPH plus H plus, oh, don't like that, plus H plus is coming along and being that little electron taxi that drops off the electron, hence the reason it's a reduction. That then produces G3P, okay, glyceraldehyde 3-phosphate. So what happens is, it then has ATP come in, it phosphorylates, so you get the second phosphate on there. NADPH plus H comes in and drops off some electrons, and that then makes a 3-carbon molecule known as G3P. Okay, that's what actually comes out. Remember we talked today about how it's a white lie in baby bio that you make glucose. It's actually you make G3P, okay? And then you can do all sorts of different things with that. Make all the organic molecules that you need. So, this G3P, okay, one of them goes out, and that's used to make all sorts of different organic molecules, right? But what happens is, one goes out, 5 go around. So, when this happens, you're going to get 1 going out and 5 of them keep going because you have 5 3-carbon molecules that are going to be used to generate 3 5-carbon molecules. So changing some partners, doing this regeneration of RUBP, okay, to be able to do that. So it's just using phosphorylation to do that, okay? So now, there is a problem though with photosynthesis in that you can have what's called photorespiration. In other words, it's where there's like an oops. So this is why there are plants that instead of being C3 plants are either C4 plants or CAM plants, okay? So, when you don't get enough CO2, because remember, you've got to be able to do gas exchange to be able to get the CO2. It's got to come in through that hole. Problem is, you're losing water. So in really hot environments, You might close those because you don't want to die, but you need the CO2, okay? So, if you don't get enough CO2, it could be you've got a partially closed stomata. Could be, you know, that's the biggest reason, right? You're in an environment where it's too hot, you're going to lose too much. So then photorespiration is where RUBP carboxylase, remember that enzyme? It would rather bind to O2 than CO2. It's more attracted to oxygen than CO2. That's a problem. So now it's dancing with the wrong partner. So then you get C2 molecules instead of this unstable C6. Screws everything up. You don't get to do the Calvin cycle now. What happens is these C2 molecules are useless. So they get pumped to the mitochondrion so they can get broken down, or they get pumped to a peroxisome, and so it gets more oxygen. That actually causes this really nasty vicious cycle so that plants can actually basically starve themselves to death. Okay, and that's a problem. So then what you've got is this process of how do you survive that? So you get this problem of no sugar, lower ATP, and you get less glucose by about 50%. Amazingly enough, most plants can survive that until you get an environment where it's too freaking hot and you're going to lose too much water. Okay? That's why, how do you deal with that? Well, you need to fix or take in more carbon dioxide if you're going to do that. So how do you do that? You can either do that, right, by being a C4 plant. Sugarcane and corn are examples of that, right? So those stomatas are only open part of the day. What that means is they're going to use more parts to get the job done, which is less efficient, but they can do it with their stomatas open less. So you're going to still be using the mesophyll cell, and it's going to use a different enzyme, PEP carboxylase. It's a modified, it's slightly different than RUBP carboxylase, and it's a different enzyme, so therefore different product, right? Enzymes are specific, they have specificity. So it's going to produce what we call C4 molecule called malate. That's why they're C4 plants. Those then get transported to, remember we were talking at the beginning about those bundle sheath cells? So it gets transformed there in the bundle sheath cell, and it gets the CO2, gets taken out of the C4, and then that CO2 that is now inside that bundle sheath cell, so it's in that bundle sheath cell, gets released as a gas, right, over to the palisade mesophyll, and so therefore you can finish the process of those light independent reactions. The other option is to be a CAM plant, crassulacean acid, I'm probably butchering that, and things like succulents. In other words, things like cacti. There again, stomata needs to be closed all the day and open at night, right? So CO2 is fixed into organic acids, specifically this one, right? That's an example of one. When those stomatas are open at night. So weirdly enough, these guys kind of like store CO2 for later, right? So these guys are sucking in CO2 at night and storing them in organic acids that during the day, right, they can take these organic acids that get stored in these mesophyll cells in the vacuoles, right? Remember, you don't just store water. And then what can happen is during the daytime, when they got their stomatas closed, they still have access to CO2 because they break up those organic acids, they release CO2 that they then strip the carbon off of to be able to get this carbon dioxide, right, that needs to get the carbon to be able to go into the Calvin cycle. Okay? So, why is photosynthesis important? Human perspective, of course, is that, of course, we like to live, so oxygen is good. Okay? We also need to get rid of those lovely greenhouse gases, right? That's one of the reasons that clear-cutting forests causes a problem. Okay? Or contaminating your seas is a problem because a lot of that lovely photoplankton is giving off oxygen that we breathe, and if you kill them off by acidifying your oceans, that's a problem. We, of course, also have food production at a certain amount of metric tons, okay? And that's even, right, think of this. This is with the respiration even happening. And, of course, that coupling in that last diagram that we did, it's the coupling of aerobic cellular respiration with that photosynthesis. So hopefully that does it. Thank you. Bye.